Hello my dear friends and my dear students, Assalamu alaikum. Friends, we are here to discuss about the tissues of the human body. Now you know that we have started about discussion about the epithelial tissues. In the previous lecture I have discussed about the structures which are present in the lateral surfaces of the epithelial cells, right? So go and watch these lectures to have a clear concept of the connections between the lateral surfaces of the epithelial cells. And in the lecture before that I had discussed, I had told you that one epithelial cell can has three important sides. That is the apical side, that is the basal side, and that is the lateral side. So lateral side may be this one, maybe this one, or on that side, that one. So we have discussed about the basal side, in which I have discussed about the structure of the basal lamina, or we call it the basement membrane. And then we, in the previous lecture, we have discussed about the lateral parts or lateral surface of the epithelial cells and what are the structures in the lateral surfaces. Now in this lecture we are going to discuss about the structures or features of the apical surface of the epithelial cells. Now let's go for it. Welcome back friends. Let us see about the structures or the features of the apical surface. So let me write here features of apical surface. Okay, so in the apical surface in humans, right, the epithelial cells, let me draw one cell here. First of all we will clear the concept, then we will go for the detailed structure. Right friends, this is one epithelial cell, for example. Right. Okay, so this is epithelial cell. Now look, in certain areas of the body, the cell membrane will project upward and will come again downward, right? In certain areas there will be projections like this, right? We will see what are these projections and how are they formed, right? Then there are, in certain areas there are same projections but little bit longer. I told you same projection, same structure, same composition, but little bit longer, right? And then in some areas of the body, in some epithelial cells, right, in some epithelial tissue, there are long extensions, right, long extensions, and these long extensions are motile, or we say they are mobile, right? They are motile. They show movement, right? Such short extensions, finger-like projections, are called the microvilli. Microvilli. We will discuss about the structure of microvilli. Then these projections, which are longer than the microvilli but shorter than these cilia, they are called the stereocilia. Stereocilia, right? And then these long motile structures are called the cilia, right? So in our body, in the epithelia, in certain areas there will be microvilli. For example, in your small intestine, right, there are projections which are called the microvilli. Then there are cilia, right, which are present mostly in our respiratory tract, we know. And then there are stereocilia, which may be present in the epididymis, in the vas afferens, in the hairs, right? So these are present there and in certain areas of the body. Now let us go in detailed structure of the microvilli. First of all I will discuss about microvilli, then the next lecture we will discuss about the cilia. So the apical surface structures will also be covered, right? So let's discuss about structure of cilia. Structure of cilia. Microvilli. It's quite a little bit easy, that's why I'm discussing it first. So this is microvilli. Now let me make here a cell and I will discuss about the structure of microvilli. So friends, this is the cell, right? We will make an enlarged diagram to clear our concept. This is a cell, right? And this I have drawn, this is the cell membrane, right? And look carefully, in certain areas, as I told you, that the cell membrane will project upward like this, right? And it will again come down. Again came down, then gone, and then again upward projection, right? Then again it will come down, right? And then in one cell there can be many projections like this, right? So what is inside these upward projections we will see right now. Okay, so these what I have drawn are called the microvilli, right? This is the epithelial cell. This is the nucleus of the cell. He is very happy because we are going to discuss about this cell, so he is happy. Now look carefully. These are the upward projections of the cell membrane, right? So first of all, we should know that the length of the microvilli can be, first they are finger-like projections, right? What are microvilli? They are finger-like projections of cell membrane, right? Then the next important point, what can be the length of these microvilli? So the length of these microvilli can be 1 micrometer, which is very, very small. So they are very small upward projections of the cell membrane, right friends? Now let us go for the detailed structure first. There is on the apical part of this microvilli, right, there is a glycocalyx waxy layer, right? So what is there? There is a glycocalyx layer, which is glycocalyx are made up of carbohydrates. So this is a glycocalyx layer on its upper surface. So we will discuss only about 2 of them, not all of them, right? So the first of all, above this on the surface of this microvilli, there is a glycocalyx layer, right? So what is this? This is glycocalyx, right? These are carbohydrates, so they are present on the surface of these upward projections. Then the first thing that we discussed is a glycocalyx layer, and what is its location you have seen right now. Then the next important thing is that we should know that here should be certain proteins which will go up and will keep these microvilli firmly in their position, right? You know, there are pillars, right? When you are making pillars, right, what do? You look carefully. First of all, I will draw a very simplified structure of a pillar. We just dig the ground. This is the ground. For example, we will, what do? We dig the ground, right? Then we put certain building substances here, right? Okay. Then what do? We take some parts of it to go upward like this, right? Like this, right? And then we again put certain building materials here to make it very, very strong, right? So this is, now cover it, right? This is the pillar, right? So same structure is found here in this microvilli. So first of all, look carefully. And there will, this is the cytoplasm, so there will be certain proteins which are present in this cell, in this cytoplasm. I will discuss about these proteins and I will write their names, right? So these are the proteins which are present. First of all, we should know in this cell, in this inside of the cell, which is called the cytoplasm, right? These are the actin proteins, right? Then these actin proteins are anchored by certain other proteins inside the cytoplasm to hold them very firmly in their position, and we don't want them to go anywhere, right? So these are the proteins which hold them very firmly inside the cytoplasm. They anchor them, right? So here is again that structure we will draw, right? I will write the names of these proteins, right? They make the base, right? They make the root for this pillar. Okay, so these red proteins are called the actin proteins, right? So let me write it here. These red proteins are called the actin proteins, actin filaments, right? Then these blue proteins are called the intermediate filaments, intermediate filaments, right? So intermediate filaments will anchor these red proteins, the red filaments, into the cytoplasm very strongly, right? So first we made the base, right? Then what do? We take some proteins of these. Look carefully. Another important point, listen carefully. Certain proteins are here which will hold this basal body or the base or the root to the cell membrane, right? These proteins, these are called the spectrin proteins, and these spectrin proteins are making strong connections between this root or this foundation with the cell membrane, right? So these proteins are called the number 4, these are called the spectrin proteins, spectrin proteins, right? We will put these there also. Okay, then what happens from these red proteins? Their fibers will go up, right, like this. Their fibers will go up, and these are, what are these? These are, again, these are actin filaments, and about 30 to 35 filaments will go up, and they will make a very strong pillar there, and this pillar is called the microvilli, like this, right friends? So again, here also I told you how many protein filaments will go upward. There will be about 30 to 35 protein filaments will go up in one microvilli, right? So like this, like this, right? Then what are these? These are the actin filaments. Then what happens? These pillars, right, these filaments are connected to the cell membrane horizontally also, right? These are connected to the cell membrane horizontally like this. Please remember all these important points. You will never forget the structure of microvilli. They are also interconnected with each other, and such type of protein is the myosin F proteins. What are these? These are the myosin F proteins, like this, right? So very strong connection has been made, right? So we say, what are these filaments which have gone upward? These are again the fifth filament. Look carefully friends, the first filaments, right? The first thing which we had shown here was the glycocalyx. Then these red filaments are called the actin filaments. Then these blue filaments are called the intermediate filaments, and then these green filaments or proteins are called the spectrin filaments. And then number 5, what are these which are going upward? These are again actin filaments, right? And what anchor these actin filament pillars to the cell membrane, right? These are the myosin F filaments. What are these? These are the myosin filaments. So the 6th one is myosin filaments, right? So in one cilia we have what? These are very important proteins which make the structure of the cilia. Very simply we discussed about the structure of the cilia, so I hope it's complete now. Now listen carefully. Look, one thing I want to show you here about the stereocilia, right? What is cilia? Cilia are the finger-like projections which are 1 micrometer in length, right? And these are the components, right? These are all the components of one cilia. One cilia has all these components, so we should not forget this is 5, right? So next thing which is very important is that, as I had told you, that if there is a little bit longer pillar, right? This is a short pillar and this is a little bit long pillar, right? Same is the structure, same is the composition, same is the arrangement, but the pillar is a little bit longer than these pillars. So such type of pillars are called the stereocilia. Again the same structure will be found, right? So firstly we draw it. What are these? These are the intermediate filaments. These are the intermediate filaments. And what are these? These are the spectrin proteins, right? Spectrin proteins, and from them there will be the actin filaments, again going up but a little bit longer, right? So these are the same proteins and the same structure there, but we should not forget that these pillars are a little bit longer pillars, right? So these pillars are called the stereocilia, right? So this is one stereocilia. Right? And what are these? These are microvilli. What is the difference between microvilli and stereocilia? That microvilli are little bit shorter, but stereocilia are little bit longer than what? Than the microvilli, right? What is the location of the stereocilia? In which parts of the body are the stereocilia found? They are found in ear, as I told you in the beginning of the lecture. They are found in the epididymis, right? And they are friends, right? So stereocilia may be found in ear, maybe in the epididymis, and they are found in the vas deferens. And microvilli mostly, you know, they are found in our small intestines, right? They are found in small intestines. This was about the structure of the microvilli and stereocilia. So we did discuss these two structures. In the next lecture I will discuss about the structure of the cilia in detail, inshallah. So friends, about the apical surface, features of the apical surface of the epithelial tissue in our body, in humans bodies and other animals, we discussed about the microvilli and stereocilia. They may be found. In the next lecture we will discuss about the cilia. So see you guys in the next lecture.